Did you know that artificial intelligence is not just predicting how long we might live, but also identifying ways to extend a health span? That is, years of life spent in good health. How close are we to using data and algorithms to make aging a thing of the past? What if the same technology that powers your smartphone's recommendations could also uncover the secret of delaying or even reversing aging? Welcome to Drug Diaries, the podcast where we explore the ideas, innovations, and individuals reshaping the future of healthcare and wellness. I'm your host, Kaushik Trivedi, and today we are tackling one of the most compelling questions of our time. How can we harness technology to unlock the secret of aging and longevity? And to help us unpack this fascinating topic, I'm thrilled to welcome Raghav Saigal. Raghav is a computer scientist turned longevity researcher who's at the forefront of using AI and computational tools to revolutionize our understanding of aging. His journey has taken him from coding to collaborating with luminaries like Dr. Morgan Levine, and now to his role at Longevity Tech Fund, where he scouts the most innovative startups in the longevity space. In this episode, Raghav will share his insights into the power of computational science in longevity research, the future of AI-driven health technologies, and the challenges of turning groundbreaking research into practical, life-extending solutions. We'll also delve into ethical considerations, exciting new trends, and Raghav's personal vision for a longer, healthier future for all. If you have ever wondered how close we are to achieving true breakthroughs in longevity or what it means for society when we do, this episode is one you won't want to miss. So let's dive into the science of living better and longer with Raghav Saigal. Welcome to Drug Diaries, Raghav.
Thanks for having me, Kaushik. And sounds like a very exciting episode, to be honest.
Exactly. Now that we have you, I'm sure we are going to have a lot of exciting conversation. So, Raghav, from computer science to studying aging and researching on longevity, that's quite a radical shift. Was there a moment or some influence in your life that sparked this interest?
Yeah, I would say it's been a long journey for sure. I would say there were again, it's been definitely sort of a wide variety of events that happened during my course from doing sort of an engineering and electronics and communication, which was a completely different field, not studying even computer science for that matter, to building a computational biology startup. But I think that one thing that really sort of struck me and sort of got me on this path was this one particular interaction that I had with Dr. Morgan Levine, who was kind of my first mentor in the field of aging. So when I first joined Yale as a PhD student, I was really enthralled by the idea of building sort of computational biology platforms to solving cancer. And my whole idea was that I want to build the next sort of this AI platform that will be able to, you put in a little bit of data and out pops a drug target. And that's kind of the future in some ways. But then I attended this kind of really interesting talk by Morgan. And it was a very coincidental interaction because I wasn't really planning on attending the talk, I'll tell you that. But as grad students, we often don't get a lot of money. So
Right.
this particular lecture had free food and we were offered free food. So I went along and Morgan was giving this really amazing talk on aging. And that kind of changed my perspective, all thanks to free food. What she essentially said was in that lecture was that instead of sort of viewing aging as a natural phenomenon, what if we view aging as a disease almost, as something that could be potentially cured? And that in some ways changed my perspective, because if we could potentially cure aging as a disease, we could potentially target all aging related diseases, including cancer, cardiovascular disease, diabetes, so on and so forth. So why not target aging instead of just simply targeting an age-related disease like cancer or diabetes or cardiovascular disease? And that kind of led me on the path of going from just building sort of simple computational pipelines to maybe solving cancer to rather overall solving aging.
I know Dr. Levine has done incredible work on understanding biological age. And what was it like working with her? And how has her research influenced the way you think about aging?
I think Morgan's influence on my work has been sort of surmountable, I would say.
Right.
It has sort of in my early years in the field, it has what has sort of driven my thinking as what is it really molded my
Hmm.
thinking. So she was one of the first people to come up with one of the first biological metrics almost
Right.
sort of, one of the first metrics for biological age which
Hmm.
could be quantified, which is known as phenoage. And the way she did this was really interesting, was she used sort of very well known clinical biomarkers and used them
Hmm.
to predict sort of mortality. And it's a very simple idea, because instead of just predicting sort of using chronological age, now you're using sort of these very well known biomarkers that change with age, and
Right.
you're predicting, associating them with mortality. And you can
Hmm.
then predict how old a person might be based on those biomarkers. Now, this very idea is really powerful. And that actually has influenced my own research, because that very idea actually led to my own project, which I worked with Morgan, leading up to building what we call sort of these organ specific ages. So
Right.
instead of building sort of aging in modeling aging in the whole body, we could
Hmm.
now model aging in specific organs the same way, using the same
Wow.
very ideas. And again, just from whole blood. And that the basic concepts of her ideas and ideologies have really inspired me and my work and moved forward the field in so many ways.
Right. And do you think aging varies from organ to organ?
I absolutely believe aging varies from organ to organ. Like,
Wow.
even before my research, I think there was proof of it, because if all of us were aging the same way, all of us
Hmm.
would die at the age of 80 with the same disease. But we don't. Some of us get cardiovascular disease, some of us will get Alzheimer's, others would die of renal failure. So clearly, we're aging in different
Different
organs at
ways.
different rates. So there is
Yeah.
already sort of knowledge of the fact that we are aging in different organs at different. But the question is, how
Right.
do we go about quantifying this now?
Right.
Till now, we just look at sort of clinical biomarkers and just try to predict the disease, like the onset of disease. But
Hmm.
is there a way to preemptively quantify the fact that, hey, this is how fast your organ is already aging?
And the idea of computational approaches in longevity research is really fascinating. Can you tell us a little bit more about how do you use computers and data to figure out what's happening as we age? And also, if you could share some examples of how this has worked in your research.
Yeah, for sure. So I think at a very simple level, right, doctors are some of the smartest people I know, right? And they can figure out trends for patients,
Hmm.
and they will be able to tell you that if your cholesterol is high, you're at a higher risk of cardiovascular disease. But now think about, they can do that at an individual level, maybe
Hmm.
even for 10 patients. But what if you have to figure out a trend for 10,000 individuals, for 100,000 individuals? How do you do that? You can't ask the smartest individual out there to look at 100,000 data points. That's where computer science comes in. That's where sort of these novel machine learning algorithms can be really smart in figuring out trends in large amounts of data, and figuring out how can is there a trend in the biological variables that we are looking at. And these biological variables could be anything. They could be clinical biomarkers, they could
Hmm.
be sort of more interesting omic biomarkers like epigenetics, the ones I study.
Right.
And if you might know, clinical biomarkers are typically in the numbers of hundreds of clinical biomarkers. But
Hmm.
epigenetic biomarkers are in the order of hundreds of thousands, even
Yes.
millions. And that's a lot of data. So, million data points per individual, and we're looking at 10,000 individuals. 10,000 cross million. That's the number of data
Right.
points. That's not something a human can do. That's not something you and I can do. That's something only a computer can do. And that's
Right.
that's where sort of the power of computer science and AI comes in.
Okay. When we look at the biomarkers at scale, are we not contradicting the concept of personalized medicine, where we treat every individual individually?
On the contrary, I think we are going to the point where we can look at data at scale and
Hmm.
look at very nuanced changes and
Right.
very nuanced differences and nuanced. So, what we can do is look at broad changes and broad trends, but we can also look at deviations from those broad trends. And by looking at those deviations from those broad trends, we can look at those personalizations. So, for example, if you are generally following a broad trend of normal aging, great. But
Right.
now, if you are not following that generalized trend of broad normal aging, that's
Mm-hmm.
where the personalization comes in. And that's where we can
Gotcha.
really go down and say, hey, is there a way to further personalize this? And that's where furthermore sort of these AI algorithms are really powerful, because they can really pick on those really small variations and very
Right.
small trends, which again often humans can miss.
Yes. And according to you, what's the toughest part about using technology to study aging? And how do you tackle those challenges?
I think it's bias in data, because fortunately we have been collecting a lot of data, but unfortunately the data always is dependent on the context it's coming from. So we need to know the context a data is coming from. So I'll give you an example. So
Mhm.
we use a lot of cohort studies. UK Biobank is a very well known cohort study.
Yes.
But a limitation of the UK Biobank that has now been known is that very often the people who go to UK Biobank and are give their blood samples are typically more wealthy, are more coming from better backgrounds, and so the data that is being collected is in some ways biased,
Biased.
right?
Yes.
And so the models that we are then building are then in some ways are potentially biased to people who are socioeconomically better, who may be potentially of a certain ethnic and ancestral background, of a certain socioeconomic background. We need to be cognizant of that. Now, again, we have data, so we should still do the modeling, but when we are doing the modeling, we need to be
Hmm.
cognizant that those challenges exist in the data and we need to account for those challenges. One of my own personal issues with the field, for example, has been that in the field that I am
Hmm.
in longevity and aging science, is that a lot of validation, for example, and analysis in the field has been done primarily in sort of Western centric populations.
Right.
There's actually little to no analysis on South Asians, which personally frustrates me. I have built a biomarker and validated it. It's been patented and licensed out, and unfortunately, there is not a single person who is South Asian in the
Hmm.
training or validation of that biomarker, just because there is no data that exists out there.
Right.
Now, finally, we got access to a data set that has South Asian, but it's almost after 3-4 years of trying to get access to this data set. But that is the state of the field, unfortunately, that such data sets do not exist. Such biases are prevalent in the field. And so one has to be very cognizant of that.
Can you give us an example of some of the biomarkers that you're tracking in aging and longevity space?
Well, they can be of different kinds, of course. There can be clinical biomarkers that are often used by physicians on a day to day basis. The limitation of current clinical biomarkers that are there is that the current paradigm of medicine is mostly of reactive nature,
Hmm.
which is that it focuses on either diseases, or it focuses on once you get a disease, we will then treat you. So the
Right.
biomarkers are built for that purpose.
Okay.
Um, they're not built to essentially be preemptive in a lot of
Hmm.
ways to prevent diseases from occurring
Right.
significantly. Even so-called sort of the healthy ranges of a lot of
Hmm.
these biomarkers are often not the optimal ranges for these
Right.
clinical biomarkers. So, clinical biomarkers are great, in my opinion, and we often use them even for
Hmm.
our training purposes, but they're not ideal. One of the biomarkers that we often sort of look at are DNA methylation aging biomarkers, or epigenetic
Right.
clocks, which have been potentially you said as sort of as potentially the next generation of aging biomarkers that could be really powerful. Why are they said to be powerful? Because they are prognostic they are predictive of future diseases, future mortality, morbidity in a wide variety of outcomes, frailty, so on and so forth. We recently
Hmm.
published a paper where they have also been shown to be potentially responsive to a wide variety of interventions, and they are also significantly reliable now. Uh,
Right.
and In the same vein of things, there are also sort of proteomic biomarkers that are coming out, sort of these proteomic clocks that do seem to be also prognostic. Their responsiveness and reliability still needs to be tested. But these are sort of these next generation of biomarkers that are being potentially made now to translate into this context.
And for our audience, could you maybe spend a minute or two and explain the concept of DNA methylation?
Yeah, for sure. So DNA methylation is really, really interesting as a biological layer of sorts. Um, so we have sort of the central dogma of biology. We have DNA,
Yes.
RNA, proteins. DNA methylation lies between sort of DNA and RNA in this sort of central dogma. And what it essentially does is these, as methylation says, it's these small methyl groups, these CH3 carbon and hydrogen groups, three hydrogen sort of group that gets added onto sort of your DNA. And what's the purpose of this group, this methyl group? The purpose of this methyl group is that the presence of this methyl group on the gene essentially prevents this gene from being expressed very excessively.
Right.
So the more the methyl group is, the less will this gene be expressed. The less this methyl group is on the gene, the more this gene would be expressed. Now, what's really interesting about sort of DNA methylation is that it can be modulated by socio-environmental factors. So, for example, stress can effectively change your DNA methylation. Pollution changes DNA methylation. Alcohol changes DNA methylation. Smoking changes DNA methylation. Biological factors, of course, change DNA methylation. For example, DNA methylation is changed by aging. DNA methylation is also what separates a skin cell from a heart cell. So all of your skin cell and your heart cell both have the same genes. How does a skin cell know that it has to become a skin cell, and how does a heart cell know how it has to become a heart cell? It's because they have different DNA methylation marks.
Right.
A skin cell knows how to become a skin cell because it has different DNA methylation marks as opposed to a heart cell. And so it plays a critical role in biology, in differentiation of cells. So it has a multitude of roles to play. And so it's kind of like what we call as if DNA itself is the code of life, it's the hardware. Then DNA methylation is the operating system. It is what controls everything. And what's really critical about DNA methylation is that it is also partly heritable. That unlike sort of genetics, which kind of have take long to get modified, DNA methylation gets modified very quickly and can be inherited from one generation to another. But it doesn't stay on for a really long time through multiple generations. It's been seen in sort of C. elegans, which is like a model organism, that it can last to up to 10 generations, for example, in certain cases. And it's a really interesting way for the body to sort of manage the genome in some ways. So, of course, the genes have to be expressed, but you have to control which genes have to be expressed and which genes cannot be expressed, or else the genes will continuously be expressed. And how do you control it? You control it through DNA methylation. One of the ways to control it is through DNA methylation.
Got it. That was very well explained. I really like the way you simplified the whole concept of DNA methylation for the audience. Now, you are constantly scouting for startups and new technologies and new ideas. What gets you excited? Are there any things that make you think this may not work or this may work? What are the preliminary signs where you smell some success and some good work going on?
I think I'm a scientist at heart. And the first thing that sort of gets me going is good science. I think if the science is BS, I think it will not be interesting. And unfortunately, the field of longevity has been one that has always also been sort of laced with a lot of these pseudoscience ideas very often. And so I think as a being in the venture capital space, one of your first steps is just to sort of weed out between the science from the pseudoscience. As a scientist, like that is the first thing I do. Like,
Right.
is the science backed? Is there actual literature supporting this idea?
Right.
Is this just made up science, or is this actually science that can be true? And then I think within the fact that if the science is true, there is the second part, which is the science actually possible to translate to patients? Because there are
Right.
so many ideas that are great scientific ideas, no doubt
Yes.
about it. But in realistic parts, are those ideas even plausible to do in practice? I don't want to give ideas out here at a public forum, but there are scientific ideas which are great in theory to do to solve aging, but I don't think they will happen within the next 20 to 30 years. So to invest in them now for us, for a fund that is looking to have a period of sort of getting an exit in the next 10 years, I don't think it's worthwhile for us. So will the science be tangibly accessible and useful to people in the next 10 years
Right.
and translate to something meaningful? I think that's the second critical question. That is the science translatable?
And are there any pathways, ideas, technologies that you're particularly excited about?
I'm personally very excited by ideas that are simple and can get the job done. Basic science and built on basic science is a disruptor idea. Now, I'll give you one or two examples here, maybe. So, I'm sure you must have heard about kidney dialysis to clean out the body of toxins in some way.
Yes.
Now, imagine if you could miniaturize a kidney dialysis into a small little sort of nano robot
Okay.
that can go into your body, suck it
Right.
up, suck up any sort of undesired
Toxins.
toxin, and get eaten by a macrophage. Simple
Yeah.
idea, really powerful. And it's completely disruptive because everything right now in longevity is focusing on adding new molecules to your body. But this is thinking about draining ill toxins from your body. And this was a company that the fund invested in, longevity tech fund. This company is called Nanotics, and this was one of the first companies I personally invested in.
Okay.
Because I was so excited by it. Because this is revolutionary. Because in my opinion, you can add molecules to the body, which are really
Right.
exciting, but they are always going to have an effect. They are always going to have some kind of a side effect.
Yes.
But the best thing is you should remove molecules from the body. And we already see effects of that. Plasmapheresis, for example, is nothing but diluting bad molecules, as we know that. Yeah, go on.
Sorry. Is it possible that the macrophages might max out because there's so much going on within your body that you may not have enough capacity to be able to absorb all the toxins?
So it's a very targeted therapy where you're looking at only, for example, absorbing IL-11, which you might have heard is a very interesting molecule that has come in to play right now, which has shown to extend
Yes.
lifespan by 35% in mice, right? So this particular nanot, as they call it, right, will essentially suck up just IL-11 and be consumed by these macrophages. That's it. And hopefully extend lifespan for these individuals and health span. And again, simple idea, simple technology. Another idea that I came across recently that was really, really inspiring to me was actually based on DNA methylation. A very basic science about DNA methylation, and that's often used by these cancer companies, is that we have these cell-free DNA, which is used by companies like Grail and so on and so forth to essentially make these cancer tests. And you have now these billion-dollar companies essentially based on this. And the idea is that what happens in cancer is these cancer cells essentially want to express as much genes as possible so that they can essentially wreak havoc in the cell, proliferate in whatever way they can. So what they do is they cause hypomethylation, which is they essentially remove as much methylation from the cell as possible.
Okay.
So they have very less methylation on it.
Right.
While regular cell-free DNA has normal methylation, or what we call as hypermethylation. So if you were to extract cell-free DNA from a non-cancerous cell or from a non-cancerous tissue or from blood from a non-cancerous person and a cancerous person, you would essentially see that hypermethylated DNA will fold up in a polar solution differently versus a non-polar solution. So a non-polar solution in a polar solution, it'll be kind of free floating, while in a polar solution, the same hypermethylated DNA or the non-cancerous one will fold up really tightly. So it'll fold up differently. And if you run a basic sort of electrical charge through the solutions, these two will have different resistance.
Okay.
And you can essentially make a biomarker out of it.
Wow. That's genius.
And that's it. You don't need something as complicated as Grail. You don't need to do NGS sequencing. You don't need to do any sequencing stuff. You don't need to do PCR, any of the stuff, just this.
Wow. That's revolutionary. Wow.
It's disruptive,
I never thought of that way.
right? And it's basic science. Basic science, disruptive technology. And that's the kind of stuff that I love investing in, that you take basic science and you build something out of it. And this company is called as Asima Health. Yes, and it was started by a really, really smart professor from actually IIT Delhi, and she's doing amazing stuff. And yeah, it's, again, an amazing company, in my opinion.
Wow, that's really groundbreaking and fascinating and powerful concept.
Yeah.
It's really amazing. Raghav, what role do you see venture funding playing in accelerating research to market for longevity tech?
In my opinion, academia is a really powerful space. And I've worked in academia, I've been in academia, I still am in some ways. And academia is an amazing space to develop new ideas. But the challenge I see in academia is the translation of these ideas. One of the biggest challenges I see is that, unfortunately, academia, the environment is not built to sort of light speed the propulsion of these ideas into translation to they need to be at where they can be given to patients, right? And partly the reason for that is you need funds, of course, and often grants don't have enough funds that can do that. Second is you need personnel, right, to do that kind of research. And the honest answer is that the biggest labs out there have 50 people. And a typical size of a lab, even at Yale, is like 10 to 20 people. And they are doing probably 20 projects in that lab. A single person is probably doing 2 projects. It's just not feasible for this to translate, for an actual project to translate within academia. So what you need is like a dedicated group of people working on this and the funds to do it. And that's where sort of venture capital can really help, right? It can help build that environment. It can provide those funds. It can help build the environment. It can provide connections to the people, right, who can help you think about how to structure translation of a really interesting and exciting academic project. And let's be honest, for a large point in time, these projects are of academic nature to an actual scientific technology, because there is a gap between academic project and a scientific technology, and a lot of things fail between that gap. And there needs to be a lot of effort that goes in, and that's where sort of venture capital comes in.
Also for fast tracking the research work.
Exactly.
And scaling it up.
Yes.
Right. Now, on the topic of potential of AI and machine learning in longevity, you partly touched upon this topic, but if you could dive a little deeper into how AI is being used to predict biological age and potential life extending interventions, and are there any specific AI-driven projects or models that you are excited about?
Yeah, for sure. So I would say there's so much happening within the AI space in this kind of last few years that has been transformative, and that has been spilling over into the space of medicine and geroscience as well. And on a very simple level, we've been using very basic machine learning from the very beginning of prediction of biological age. So
Right.
we used the very first models of predicting biological age use something as elastic net, which is a very basic form of
Right.
a machine learning. I wouldn't say a neural net model, but a very basic form of a neural net model. And we have since then evolved, and we have used more deeper neural net models that leverage and use more complicated models.
Right.
Recently, we came out with a paper called CPGPT, where I was a co-corresponding author, which uses the same architecture as ChatGPT, sort of the transformer-based architecture, to predict mortality and a bunch of other this thing from epigenetic data and DNA methylation. So the field is growing and burgeoning with all of these AI innovations. And the basic idea is that the complexity of the data, right, as I mentioned, is so huge that simple models are limiting. And so
Yes.
you need more and more complex models to understand better about the data. And the better and more complex model you have, the better you will be able to understand about your data. And that's the crux of it, that the better your model is, the more complexity it can understand, the better you will be able to predict and understand about biological age. And that's it. Now, how is this being? So there's one component of understanding biological age. You build kind of these machine learning models, these AI models, right? You use epigenetic data, proteomic data, clinical biomarkers, whatever kind of data you use on thousands, hundreds of thousands of patients, right? On lots of features.
Right.
But the other point is, at the end of the day, you want to have an intervention, because this is all in the goal of having an intervention. So let's talk about how do we get there. So the first thing is, well, building a better biological age biomarker is super helpful for having interventions. So that's one clear way how this would be helpful. Using AI to build a better biological age biomarker is
Yes.
super helpful to get to interventions. And the reason for that is simple, that if if we were to currently run a clinical trial for a interventional study for longevity, it would last 30 years. The
Right.
reason for that is the endpoint for a clinical trial like that would be mortality or death, because what we are really looking for is extension in lifespan.
Yes.
But we cannot run this clinical trial. I don't think any country in the world has an FDA or NIH that can fund such a clinical trial, let alone a company.
Yes.
So what can we do to solve that? What we can do to solve that is essentially have proxy biomarkers. What these proxy biomarkers can essentially do is they can tell us that, is the intervention working or not? Is it being responsive? Is this decreasing epigenetic age or biological age? Because these proxy biomarkers are prognostic, they're responsive, they're reliable, they can predict that they are mortality and stuff like that. So we know that they work already. So they can essentially, within a very short period of time, they can tell us that, hey, this intervention is working or not. So
Yes.
instead of running a 30-year long trial, we can finish a trial maybe in 2 years by seeing a change in these proxy biomarkers. So that's one way. By building a better biomarker using AI, we can sort of help interventions. But the second way we can help solving new interventions using AI is kind of building new platforms and new technology. And so that's where the really sort of next generation stuff comes in. So we have these really new technologies, really cool technologies that are coming about, where we are now able to model how a single gene or drug perturbation can affect a single cell in silico.
Right.
What that means is that if you were to give a drug to any cell in the body, what would its response be? So we are able to do that at the single cell level. I see in the next few years, we'll be able to do that at the whole organ level and eventually the
Right.
whole body level. So imagine a point in the future where you can effectively say, I want to give this compound
Hmm.
to an hypothetical in silico individual on my
Right.
computer. What would its response be? Would it
Right.
prolong life? Would it be toxic? Would it cause kidney damage? Would it cause liver damage? Would it cause cancer? Or would it be actually helpful? Which genes will be perturbed? All of those things done in silico.
Hmm.
Right now, we can do it at the single cell level, and we are
Yes.
able to do it. But the fact that we are able to do it in the single cell level, I don't think we're far from doing it at the organ and hopefully in the future at the whole body level. I see that happening probably by the end of the decade, maybe.
Wow. That's really interesting. And I read about this organ on a chip technology where you can almost create the organ on a chip and run your clinical trials and experiments and see what the outcome might look like.
Yeah. And that's again sort of another version in some ways of this technology where you're trying to essentially see what kind of perturbations that we can do in theory in vivo, right?
Yes.
In humans, but instead doing them essentially in vitro in some ways,
Yes.
but getting more and more close to the actual in vivo simulation.
Wow. That's very interesting stuff. And when it comes to AI, what are some of the limitations or ethical concerns around using AI to predict longevity or health outcomes?
So I pointed out one of my biggest concerns is kind of the training of the data. That the data that goes into training can be a huge challenge, and we have seen this already in some ways when we have done this for facial recognition algorithms. We have done this for a bunch of other things. Uh, AI can get biased based on the way the type of data that is provided, and that is concerning.
Right.
And as researchers that are going into this field, we need to be very, very cautious about this. That if we do not fix this, this will be a huge problem. For example, a biomarker that is built primarily for socioeconomically wealthy populations might not be really good for other groups. But actually, that might actually mean that would actually mean a loss of capital for companies.
Yes.
If you take that to market and it fails in a clinical trial, that's bad.
Yeah.
That's just bad. From an ethical consideration, it's bad, but from also from a capitalistic consideration, it's bad. So I would say it's a win-win for everyone if we fix some of these data issues while we're thinking about. That's one of the ethical issues that I think about a lot when I'm thinking about AI. I think another ethical issue in context of AI could effectively be the very black box nature of these algorithms,
Yes.
which is that till now, a lot of the choices that we make around health and interventions, we understand them
Yes.
as physicians, as scientists, because have a mechanistic understanding very often about these biomarkers, about these interventions. But as we use more and more AI, we may stop having some of these mechanistic understandings, which may be a bad thing, but may also not be necessarily a bad thing. I would say that
Right.
because there used to be a time. I will say this. I don't know if you have you heard about the E-rooms law by any chance?
No, I haven't.
I'm
No.
sure you must have heard about Moore's law.
Moore's law, yes. Yeah.
Yes. So Moore's law essentially states that you have decrease in cost of goods, specifically semiconductors, as exponentially. Edom's law is the inverse of that and has been going on in the pharmaceutical industry essentially, where the cost of making a drug essentially and getting it to market has significantly gone up from the 1970s to 2020s. And there are multiple reasons one could justify for it. It's inflation, it's yada yada yada. But one of the potential reasons has been the hyper focus we have on mechanistic understanding, which is quite contradictory to our understanding of science till now. Their justification is, and it's an interesting justification, I think about it a lot as a scientist, is that back in the day when therapies were tested, they would throw compounds in a petri dish and see what works, and then move it to the next stage, and then throw compounds in mice and see what works, and then move it to the next stage, without really focusing on how it's working. But instead now we go into this super long stage of being like, oh, exactly, we need to know how this works. Like, is it changing this gene or that gene? And does it have that effect? And we spend a lot of time and investment into this thing. And maybe it's useful, but maybe it's not really, and it's spending a lot of money in that stage, but is that really revealing something valuable? Right? Has that really improved our value in the odds of getting a successful drug to market? I don't think it has really changed our odds of getting a successful drug to market. It used to be 10% back in the day. It is still 10% now. So has it really changed?
That's a good question. So the incremental investment in doing all those investigations, is that leading into incremental benefits? That is questionable, right?
Yes.
So a lot of data being generated.
It's a lot of data being generated. And as someone who works in comp bio, I love the fact that we're generating so much data. It's super valuable to me. But the question is it valuable to the people that need the drugs? Is that decreasing the cost of the drugs? And maybe that's where AI can be really powerful, because AI agnostically, using just the data, using just the trends it understands, will predict us something. And maybe it's not able to explain everything, but maybe it predicts something that is way more powerful, right, than simply saying that this mechanistic understanding is better versus this thing works better. Maybe just us trying to mechanistically understand everything is not the simplest solution to this. Maybe just things are so complicated in this context that we can't understand these things mechanistically. It's kind of like the same problem when looking at 10,000 features, right?
Yeah.
There are so many features in a cell, right? We're looking at 20,000 genes, I don't know, 100,000 proteins, how many different variations of those proteins, how many different sites of DNA methylation. It's almost impossible to figure out all the states. Maybe we are not experts at figuring out the mechanistic understandings. Maybe let AI lead to the right answer.
Yeah, that's true. And in your opinion, how close are we to seeing some groundbreaking innovation in longevity tech?
I think we're already hitting it in some ways. Like, we're seeing sort of different avenues almost. So I see different ways in which we will see sort of longevity technologies being advanced. So I feel that there would be three kinds of technologies. There would be ones that would slow down aging, ones that would stop it, and the third kind that would reverse it. A lot of focus that we have right now are on technologies that are typically slowing down aging. These technologies include sort of current medications like metformin, rapamycin, so on and so forth. These are therapeutics that are slowing down aging. They are not reversing aging, they are not stopping aging in any way. We may have technologies that are stopping aging. For example, stopping aging would be something like cryotherapy, something really out there. I don't know if cryotherapy will work or not, but maybe. And then there would be something like reversing aging. That would be a full body epigenetic rejuvenation or multiple organ replacements. Now that would be like sort of, I would say, rejuvenation in some ways, right? Or really like slowing down or reversing aging in some ways. And so on the slowing down part, I would say we've made a lot of progress. We have sort of interventions that are actually going into trials now. There are different companies that are taking interventions into trials. We are already seeing interventions like semaglutide that might be multi-purpose interventions that would hopefully be longevity focused as well. We're seeing companies like BioAge coming up with their interventions that, even though they're targeting it as a metabolic disease intervention, we know that they might be sort of generally longevity focused. There are these interventions that are, as we say, are really powerful at slowing down aging.
Right.
About the stopping one,
And
I'm not quite sure. I don't know where we are. The cryotherapy, that kind of stuff, I'm a little unsure about that. The slowing down one, on the other hand, epigenetic reprogramming is really powerful. We are seeing some really exciting stuff on that front. We have recently had papers where they've been able to show that reprogramming in mice is able to rejuvenate their cognitive abilities, and also that they did a pretty harmful experiment, which is crush the nerves of these mice, the optic nerve of these mice, and then gave them these reprogramming factors, and the optic nerve built up again, and the
regenerated.
mice became not blind. So from being blind to becoming not blind. So clearly they are making significant progress in this kind of regenerative rejuvenation technologies. So I would say that stuff is a little further away, say 20, 30 years away to actually getting to humans. But
Right.
I would say it's moving again at lightning speed.
Right. And where would you place the use of genetically modified or unmodified natural killer cells in this whole value chain?
Did you say genetically modified and unmodified? Could you repeat that?
Natural killer cells.
Natural killer cells. Those could be really
For
interesting, I think. Yeah.
yeah, they are more for stopping the aging or slowing down.
I think they would be interesting in context of, like, for example, preventing cancer
Yeah. Senescent cells
or
yeah.
senescent cells or of the sort. So I would say that those would potentially come at kind of like the slowing down aging process, not so
Right.
much
Okay.
as like reversing it or
Or
stopping it, right? Because it would still lead to progression. Because even if you are able to sort of improve sort of your natural killer cell count or have them become more powerful in some ways and sort of be more active, you'll still have other components of your body or other components of other
Yes.
dimensions of aging that are still still sort of accelerating. So
Exactly. Yeah.
yeah, so you still have sort of that imbalance, and that might still give you sort of aging related diseases.
Yeah, I totally agree. I think it may help in slowing down the progression of liver fibrosis or Alzheimer's or Parkinson's, but it is not going to reverse it. It's not
Yeah.
going to reverse aging. How do you see partnership between academia, industry, and government supporting the translation of longevity research into real-world applications?
So I've been really excited about the fact that sort of government has shown a lot of interest in the recent years, sort of especially in sort of longevity research. So till now, I would say we had NIA, which is National Institute of Aging, but unfortunately, National Institute of Aging was primarily focused on Alzheimer's. Or one could even sort of maybe I shouldn't say this out loud, but I think the joke was it should be rebranded as National Institute of Alzheimer's, because that's predominantly where all 60% of the funds of NIA used to go to Alzheimer's and not to basic sort of research in aging. And that was disappointing because if you really need to move the needle on aging, even on Alzheimer's or cancer, what you really need to fund is aging science and not all these diseases of aging. But more recently, we have seen the rise of organizations like ARPA-H, which is kind of like, if you might have heard about DARPA, which was built to sort of really build up sort of the defense profile of the United States back in the day. ARPA-H has been built with the same idea in essence, but to build up the health ecosystem of the United States. And their whole idea is what they're going to do is they're going to work on technologies that are going to translate. They're going to essentially invest billions of dollars, and they're going to work on translating these really next generation technologies to clinic. And they're going to essentially make it run like a startup completely. So they're
Right.
going to spend like millions of dollars on a technology. They have a project. They essentially have these program managers that are kind of like these entrepreneurs in residence that drive these projects with critical deadlines. I love the program. And it's run like essentially like a large venture capital fund of sorts. And it's a completely different completely different way of running a government funding organization. Personally, one that should be adopted by multiple countries, in my opinion, because it's fast, it's driven by mission and translation, and the output is simple. That does the technology translate to patients and enable patients? If not, cut it out. That's it.
Right.
And that, in my opinion, has really helped the longevity industry, in my opinion, because the programs like ARPA-H, for example, they are the ones that are really thinking about preventative medicine. And that's where the longevity industry really comes in. If you really have to
Yeah.
transform the current American healthcare system, which is really in all ways reactive, and you want to have a preventative healthcare system, what you really need is the longevity care and the longevity tech industry to step in. And that's where ARPA-H has really been, in some ways, a great collaborative partner for this industry.
Absolutely. I think it's really encouraging to see that governments are playing an active role and waking up to this revolution of aging and longevity tech. I recently saw this post from Professor Neer Barzilai with the Deputy Prime Minister of Singapore. And it's really encouraging to see that the Singapore government is actively fostering research and development in the longevity space and promoting it. So
I
I
have to
think we are
say,
seeing some.
No, I have to say, Singapore government is again an amazing model. I have to say that in aging research, like they have set up some amazing, like for example, if within NUS they have like amazing sort of the aging center there, but even within beyond NUS, they have like this A-star agency, which where they are collecting about data on 100,000 individuals. Singapore is a small country. To collect data on
Yes.
100,000 individuals,
100,000. Yeah, that's a
right?
huge number.
It's
Yeah.
large. They already have data for 10,000 DNA methylation, like 10,000 individuals with DNA methylation data. That is humongous. I have
Yes,
never
huge.
seen such large DNA methylation data ever in my life. Like there are studies that have like 20,000 individuals, but never from such a small country or from such a small
Yeah.
place. It's amazing what they're doing. So I would really want like other countries to follow suit.
Yeah. Absolutely. I'm sure in the coming years we are going to see more active role being played by the government to promote and encourage research in this area.
I would say the governments have to play a role almost, because it's gonna be, I feel like it's, and this is kind of my personal opinion in this context, is if the governments don't understand the need for longevity science and the need to pivot, it's gonna be a huge backlash on them. Western economies
Yeah.
are aging and they're aging very quickly. And that is going to be a huge, huge load on their economic and social systems. We already know that things like Medicare and Medicaid are already stretched in the United States. And if they continue the way they are, they're going to run out of funds by 2030, 2035. So they need to change their paradigm of
Yes.
medicine. They need to go from a reactive paradigm to a preventative paradigm so that they can provide for their individuals better. And the same is true for sort of in the UK. We know that you guys face such long lines
Yeah.
and queues, and that's
Absolutely.
partly because of the structures and systems in place. It is this kind of reactive paradigm of medicine that is not working for economies and countries across the globe.
Yeah. I was listening to this webinar recently, and I was listening to Professor Andrew Scott, who was apparently giving a session in one of the universities in China, and he was talking about the longevity research, and in the coming few years, we are going to see a great breakthrough in this area, and people might live up to 100 years easily. And what are your thoughts? So the students said they were not too excited about it. They said the aging population is a burden on the economy, it drains out all your resources. And then he said, hang on a second. We're talking about you guys. It's going to be you.
Yeah. Yeah. No, and that's absolutely true. And here's the thing, I'm worried for myself, right? Like,
Yeah.
eh, I'm 30, right? I don't know how old you are, Kaushik, but like in the next 30 years, we're going to be 60 and 70. So
Exactly.
we're going to be the old guys. So we better start
Yeah,
fixing things now, or else it's going to be bonkers for us.
exactly. We are talking about us. Right. Uh, can you think of an example where a promising intervention faced some unexpected hurdles during practical implementation and didn't pan out the way they were hoping it would?
Promising intervention face practical hurdles. There's so many
Hmm.
in this context. I was just actually thinking about an intervention that has been like potentially very hotly debated in this context is HRT for women for
Hmm.
longevity
Right.
purposes. It's in some small clinical trials, it's been shown that it's super helpful. In other clinical trials, it's been shown that there was a WHI Women's Health Initiative clinical trial with 5,000 women almost that had very mixed results. We
Hmm.
still see the use of this in current scenarios in
Right.
clinics, and we still see that sometimes that leads to increased sort of cancer, reproductive organ cancers for women. At one point, it was propelled in the Western countries to be as the therapy to take for early menopause, but
Hmm.
now it's potentially shunned in a lot of ways, yet in a lot of cases it's still given. So I'd say that's one example. Another example I think about a lot is about supplements. I think supplements are important, but
Hmm.
when you need them. So, for example, vitamin D is an excellent supplement if you have vitamin D deficiency. It will
Yes.
help you extend your lifespan, your health span, so on and so forth. It's
Right.
really helpful for all of those purposes. But at the same time, unfortunately, vitamin D is kind of given as this multi purpose therapeutic to everyone. It's just
Hmm.
prescribed. You can get it over the counter in the United States. And in actual clinical trials for the general population, it has shown to have
Hmm.
no benefits. For
Really?
vitamin D deficiency, it's shown to have benefits. But regular for regular individuals, it has shown to have no benefits. On the contrary, there are negative issues with using vitamin D. And is that, for example, over the counter vitamin D in the United
Hmm.
States is available in what we call as extra strength, which is over 5,000
Yes.
IU, right?
Yes.
And extra strength vitamin D can actually be toxic. It's
Oh my
damaging.
God.
It's
Really?
actually damaging to people if you are not deficient. And
Yeah.
people take that, and that's harmful. And
Wow.
so there is a huge lopside to sort of even propagating some of these in sort of these supplements, right? Like we
Right.
pop these multivitamins on a daily basis, but are they really necessary? Is the
Hmm.
question. Many of them don't even get absorbed by our bodies.
Yeah. Yeah.
Um,
It's funny that you talk about this because I was recently having a conversation with one of my colleagues, and he said he buys these vitamin D shots, which are like 1,000 milligram strength, and he takes like 6 shots over the course of 6 days, and that lasts for about 6 months. And so we're not sure how, to what extent these shots are safe. Like you said, it can be toxic consumption of vitamin D.
and here's the thing.
Right.
I'm sure they are beneficial to someone.
Hmm.
I think they need to be given to someone who is deficient. I think it's
Yes.
not correct if you're not giving it to someone who's deficient. But to someone who's
Hmm.
not deficient, if you're giving it in excess, it's harmful. It is shown to be
Right.
toxic. So and people just pop them like candies. So
Yeah.
it's and so and there are a lot of these kind of, unfortunately, these we see these trends almost, right, with
Mm.
longevity and longevity science, right, and with health science in general. right? Which I often am very skeptical about as a scientist. I think the other day I was talking about this with friends. Maybe I'm going to sound like a conspiracy theorist now. But bear with me for a second. So there's a whole huge this thing that essentially in American culture, and this may be true or not true, but companies that made sugar products, right? Pepsi, Cola, all of these companies and other sort of aligned, knew for a long time that they their consumption, excess sugar consumption, caused the likes of cardiovascular disease and diabetes.
Right.
But instead, they pushed the notion that it is fat, right?
Cholesterol is the evil.
Fat is the evil. Fat is the evil, not cholesterol.
Yeah.
Fat is the evil. So what that led to was you had the whole fad of low fat diets, like low fat
Yes.
food. But what that essentially led to is because this food was low fat, so you had to balance out the taste. And to balance out the taste, you had to increase the flavoring with other things, which is sugar and salt. And two things that effectively further increased your risk of cardiovascular disease and diabetes. And so that even further worsened the cardiovascular disease risk. And you see that's happening again now with something called as gluten-free food.
Gluten-free, yeah.
Right? I'm sure people have gluten-free allergies, but the extent of gluten-free food available on the market and sold en masse is not in sync with the amount of gluten-free allergies that we have in this country. So, and what happens is because this gluten-free food doesn't have gluten in it, so it misses some of those key carbohydrate tastes, you balance it out with higher sugar and higher salt content, right? Making it worse for the people that are eating it.
Yeah, and I see that nowadays gluten-free has almost become synonymous to healthy food,
Exactly.
which is not true. If
Which
you
is
don't
not
have
true at
gluten
all.
allergy, you don't
Yes,
need to consume gluten-free food.
absolutely true. Right. It's synonymous with healthy, but is it actually healthy? It actually, if you look at the contents of the food, it has higher carbs, higher salt, and higher cholesterol content in it. So
Exactly.
it's debatable if it's actually good.
Yeah. And what are your thoughts about the supplements like resveratrol or NMN? We hear Dr. David Sinclair actively advocating these supplements. Do you have any views on that?
Well, I think my views on it are as simple as this. Unless tested, I have zero recommendations.
Faith.
Show me clinical trial data, and I would be more than convinced that it's excellent work. And I feel like they had some data in dogs that showed some positive benefit in some tests, which was interesting in my opinion. But I have not gone too much into the literature personally around NMN and resveratrol myself
Yeah.
to actually say that one is it the best this thing or not. In my this thing, as I said, both of those are supplements. And again, as I've said about supplements, my opinions are in my own research, I've not seen supplements being sort of as great interventions as lifestyle interventions or potentially pharmacological interventions. So we looked at 51 different interventions, of them about that decreased epigenetic age, of them about 14 were lifestyle, 15 were pharmacological, 11 were supplements. And the supplements didn't have much of an effect on epigenetic age.
No.
Pharmacological and lifestyle had much of an effect. Resveratrol and NMN weren't two of the supplements, they weren't there. But in general, I'm telling you, the supplements did not have much of an effect. Now, again, as I said, supplements need to be taken in the context that they should be, and I'm sure they would be really beneficial to the people who need it. Beyond that, I cannot comment much beyond that, I would say.
Right, right. Okay. What advice would you give to other researchers who want to work at the intersection of computer science and biology?
Come and join. There is so much data here. There are not enough of us. There is data by the trillions, I would say. And I guess my advice to you would be if you're bored and if you are out of luck and if you are sitting in a wet lab and you're trying to do an experiment and you have 5 minutes of time, go search for an interesting data set online and download it and write a basic line of code, and you will find an interesting insight. I am telling you that. That is how easy it is, and that is how interesting it is.
That's a very practical and wonderful piece of advice.
No, it is honestly as simple as that. I will tell you. Most of my job on a day-to-day basis as a PhD scientist or as someone who used to do all of this used to be like going on these publicly available websites and these papers and just going like, hey, this is a really cool data set. Let's download it and let's crunch it. Because
Right.
like the 51 intervention studies that we did was
Mhm.
essentially publicly available data. They were publicly available clinical trial and interventional studies. That's all they were. That's it.
Raghav, if you could solve one major question or problem in longevity research, what would it be?
I think, and this is coming from a very personal experience here, is data access.
Right.
And this is very contradictory to what I said just right now, a second ago. So, so there is one point around sort of being able to download all of this data, right?
Right.
And it's amazing that we are able to sort of have access to a lot of these public data sets. But at the same time, some of the best data sets that are out there, right, around interventions, around sort of longitudinal studies, all of the large scale data sets around thousands of individuals, right, with the
Right.
most variety, they are hidden behind these walls. I would say there is enough data in this world right now to build the best biomarker we can and potentially discover one of the best longevity interventions we can. I think we're not able to do it because we are not able to pull all the data together in one place. I think it is a limitation on us as humans to collaborate.
Got you. Now we're going to get into some rapid fire questions.
Yes.
Right. A book or a paper that deeply influenced your work.
Oh, that has to be Steve Horvath's first DNA methylation paper, where he wrote about sort of the Horvath clock. You have to read it because it is an amazing paper. But most importantly, I love the paper because he was the only author on that paper, just FYI. And because he
Right.
was the only author on that paper, he got to write it in this format.
Oh,
I
really?
did this thing instead of writing like we created this
Ah,
algorithm. He wrote, I created this algorithm and I
wow.
tested it this way. I don't think I've ever seen a paper written like that, and that's the most amazing thing I've ever seen, which is why I love that paper more than anything else. It's a seminal paper in the field, but I love the paper because the way the abstract is written.
Wow. And that is scientifically or technically acceptable. Yeah, if
Yes.
there's one individual
Yes.
who's worked on the paper, then
It is
yeah,
a
by all means,
seminal
they
paper.
would
It
get
is a
all the credit.
seminal paper that single-handedly defined the field of epigenetic clocks. And
Wow.
the way, I think partly because the most amazingly way it has been written.
Wow. That's uh, something new I learned today. I mean,
Yeah.
many things, out of many things. Technology you think will change the future of healthcare?
Technology, I think, will change the future of healthcare. Oh, this is a tough one. There are so many. Maybe I'll go against the present paradigm of who has gotten elected. I think maybe vaccines against cancer would be really powerful.
Yes, I think that sounds really promising.
I think vaccines against cancer would be superbly powerful. It's something on the lines of sort of the NK cells that you were talking about, or sort of
Yes.
hyperactivating our immune cells. But sort of vaccines against generally diseases could effectively be really powerful. Aging
Right.
diseases.
Right. Okay. The most exciting breakthrough in longevity you've witnessed recently?
This exciting breakthrough I've been witnessed at longevity recently. Can I be biased about this? The most exciting breakthrough I witnessed in longevity recently was my own paper. Yeah, I'm being very biased about this, but this is touting my own horn. We compiled sort of these 51 longitudinal longevity interventions and looked at which like at 110 aging biomarkers and
Right.
tried to see which of these interventions are actually slowing down aging in these biomarkers, right, in a systematic manner. This has never
Right.
been done before. So we can do a head to head comparison now with which biomarkers are actually working in clinical trials first, so that we can actually use them in clinical trials to kind of solve the first problem that I was talking about, right? So now we don't
Right.
really need to have these 30-year long clinical trials. But
Mm-hmm.
secondly, we can now start actually having some sense of which interventions are actually working potentially.
Mm.
Because we, till now, no one's been able to do a head to head comparison of these interventions. Everyone's been
Right.
kind of running left, right, and center saying, like, my intervention works best because it changed aging with this biomarker, and my intervention works best because it changed aging in this organism. But hey, it only matters when you can measure it on the same scale. That's what we were able to do. And
Right.
being able to equalize the scales and equalize the stage and do that
Mm-hmm.
analysis, I think it's revolutionary. We're at 51 interventions right now. We're expanding to 100
Oh, wow.
interventions and 3,000 plus biomarkers. It's going to be one of the biggest studies that is out there in the context of longevity interventions and biomarkers.
Wow, that's really impressive. And I think I wouldn't say you're biased. I think it truly sounds groundbreaking and very exciting.
Thank you. I appreciate it.
Your top wellness or longevity tip for everyday people?
Sleep well. I did not
Hmm.
realize for a very long time how important good sleep is. Eating healthy and exercising, all of those things are great, but sleeping well, nothing beats it. I personally feel like a good night's sleep can
Hmm.
be revolutionary.
Right. Of all the people listening to this episode right now, I think I'm the one who needs this advice the most. Perennially sleep deprived.
You and me both. I a PhD student here working two other gigs. You and I can
Wow.
relate to it. So, which is why I've been working on my sleeping habits. I think ever since I've
Yeah.
tried to sleep 8 hours a night, it's been life changing.
Wow. Wow. That's amazing. I got to work on it. I got to work on my sleep habits.
We'll share notes.
Yeah. Wow, that was such an amazing conversation, Raghav. I really enjoyed every second of this conversation. And this brings us to the end of this episode, Raghav. I want to thank you from the bottom of my heart for joining us today and sharing your incredible journey, insights, and expertise. You've given us so much to think about, and your work at the intersection of technology and longevity is truly inspiring. And it's clear that you're not just advancing science, you are also opening doors to a healthier, longer future for all of us. So thank you for taking the time to be here and sharing your passion and vision with our audience. And to our listeners, I hope today's episode left you with some new perspective on how technology is reshaping what it means to age. And if you enjoyed this conversation, don't forget to subscribe, share this episode with others, and leave us a review. And we would love to hear your thoughts. And before we go, Raghav, if you could share your contact details or where people can find you or get in touch with you.
You can reach out to me on Twitter, RV_Sehgal, or Instagram, same link, RV_Sehgal, or on LinkedIn, Raghav-Sehgal-Yale, or my email, raghav.sehgal@yale.edu. Happy to talk to anyone.
Thank you so much, Raghav. It was an incredible conversation. I've really enjoyed speaking with you today.
Thanks for having me, Kaushik.
Thank you for your time.
Really fun.